Chapter 10, Muscle Tissue, Part 4. All right, so back to our story. Well, of course, after the muscle has contracted, it will need to relax. We do not want to be in a state of rigor mortis all the time. So the termination of the signal, well, when the nerve impulse stops arriving, then no more acetylcholine is going to be released into the synaptic cleft. Then an enzyme, acetylcholinesterase, will come in and break down the acetylcholine found in the receptors, thereby freeing up the receptors, and this will lead to the closing of the ligand-gated ion channels, and so no more end plate potential is being generated, which leads to no new muscle action potential being generated. In the sarcoplasm, basically the sarcoplasmic reticulum, when it stops getting a signal, a charge change to open its voltage-gated calcium ion channels will stop releasing calcium, and those voltage-gated calcium ion channels will close. However, we still have the calcium ion pumps active going on, and they will continue to pump calcium back into the sarcoplasmic reticulum. Eventually, enough calcium will be removed from the sarcoplasm that the muscle contraction will cease. As the calcium ions drop, this causes troponin to no longer have calcium bound to it. It returns to its original shape. When it does that, it moves tropomyosin. Tropomyosin then covers the myosin binding sites. That will then prevent the cross bridge from being formed and prevent any further contraction. And this will lead to the return of the sarcomere back to its original length. So this relates to the elasticity of the muscle fiber. It can return back to its original size and shape. So again, a complicated story summarized in this figure that is hopefully still in your textbook, involving many membrane proteins, sodium potassium ion pumps to give us our resting potential, calcium ion pumps to trigger the release of the neurotransmitter and to trigger the contraction. Ligand-gated sodium ion channels in the motor end plate, ligand-gated potassium ion channels in the motor end plate, the voltage-gated sodium ion channels on the sarcolemma and in the transverse tubule, and the voltage-gated potassium ion channels, that should say potassium ion channels, also found in the sarcolemma and transverse tubule. All right, leaving the realm of the molecular and microscopic and starting to head to bigger structures, we have something called a motor unit. A motor unit is basically all of the muscle fibers that are controlled by the same single somatic motor neuron. So here we have the somatic motor neuron 1, follow it out, it breaks, diverges, branches out, and leads to the ability to connect these two skeletal muscle fibers. So that is one motor unit. Second motor unit made by this second somatic motor neuron, it branches out and connects to these 4 skeletal muscle cells. So that is a motor unit, single motor neuron controlling a selection of skeletal muscle cells. All right, skeletal muscle metabolism. Skeletal muscle, there are 3 ways that a skeletal muscle fiber can generate ATP. It can be super fast, immediate from the phosphagen system, basically the ATP that's already ready to go. Short-term supply from anaerobic cellular respiration, inefficient but still able to generate energy. And then long-term by aerobic cellular respiration using the mitochondria. That's our gold standard. So immediate phosphagen system. Basically, there is ATP already present in the sarcoplasm, and it can be used by the muscle fiber. So it can be broken down and used. So there's probably enough ATP in the muscle fiber for about 5 seconds of contraction. How do we regenerate it? Well, there are kinases. Myokinases will transfer the phosphate from one ADP to another ADP to give us ATP and AMP. So adenine diphosphate becomes adenine triphosphate. The other adenine diphosphate becomes adenine monophosphate. We also have creatine kinase, which is specific to skeletal muscle tissues. That can do sort of a similar thing, but in this case, it's taking a phosphate off of creatine kinase and putting it onto the ADP. So that's how we generate more ATP, phosphates removed from creatine kinase, turning it into just creatine. And these processes can be reversed. Then we have our short-term supply. Short-term supply anaerobic cellular respiration is from using fermentation, basically. We break down the glucose into the pyruvate to generate 2 net ATP. And being as no oxygen is present, the pyruvates will then become lactic acid, which will then be sent out in the bloodstream. So glucose being used up quickly, inefficiently to give us 2 ATP. That's sort of the medium short-term supply of ATP by the muscle. Pyruvate will then, because it's now lactic acid, will go into the bloodstream, end up in the liver where it'll be turned back into glucose, gluconeogenesis. So that's the fate of pyruvate via anaerobic cellular respiration. But long-term, best approach, of course, is aerobic cellular respiration, where the glucose gets broken down into pyruvate, yay, 2 ATP, and then enters the mitochondria to go through the whole process that leads to a net gain of 34 ATP, good old oxidative phosphorylation. It turns out that we can generate energy, utilize the aerobic cellular respiration by using triglycerides. In that case, the fatty acids in the triglyceride are broken down and piece by piece, carbon by carbon, removed to enter the aerobic cellular respiration in the mitochondria. So the longer the fatty acid, the more energy, the more ATP that can be generated from it. And glycerol itself, that holds the 3 fatty acids together, can also be used to make ATP. And muscles much prefer to use triglycerides because it has more energy in it than glucose. All right, so the basic idea is that in isolation, the muscle can do a quick sprint using the phosphagen system, the immediate reuse of ATP. It can have a longer sprint, 400-meter sprint, say, if it's also using anaerobic respiration as on top of the phosphagen system. And then you can have a massive run if you get a combination of all 3, phosphagen system, anaerobic cellular respiration, and aerobic respiration, because aerobic respiration is, of course, gives us the most energy possible. So in a normal muscle fiber, they're using a combination of these 3, but we just talked about them in isolation to try to be a little more clear. Oxygen debt. Well, after activity, after the muscle has used its oxygen, it needs more oxygen. What? So oxygen is normally replaced by hemoglobin in the bloodstream. Hemoglobin arrives, oxygen leaves the hemoglobin, enters the muscle cell, and binds to myoglobin. So oxygen is stored in the sarcoplasm in the myoglobin protein, very similar to hemoglobin protein, and it is actually the myoglobin protein that is red and makes the muscle tissue look red. We also need to replenish the glycogen, where we're getting the glucose. That's, of course, by eating, we get more glucose that can then be brought into the muscle fiber. ATP needs to be regenerated, and, of course, lactic acid needs to be converted back into glucose by the liver. So basically, as the muscle works harder and harder, it's using up its oxygen resources, and then eventually those oxygen resources will be replenished. All right, a contraction. Muscle fibers, there are different kinds. They generate different powers of contraction, different speeds and duration. And this can relate in one level to diameter of muscle fiber. The wider the skeletal muscle fiber, the more powerful the contraction. And that is why people's muscles get bigger when they engage in a lot of weightlifting, for instance. There are 3 kinds of muscle fibers broken down by 2 categories. We have the fiber types. This is, for instance, fast fibers or slow twitch fibers. Fast twitch fibers have a faster myosin ATPase. So the myosins are actually able to initiate and change shape faster. They can return and be ready to lead to more contractions, faster, fast twitch. So the contraction is for a shorter duration, but it is a stronger contraction because it's more powerful and faster. Fast twitch, short duration, strong and powerful. Slow twitch fibers have a different variant of the myosin, ATPase, and that means it takes longer for it to change its shape to lead to the next part of the contraction. So this leads to longer, slower contractions that are not as powerful. Slow twitch, longer contractions, but not as powerful. Then, of course, we have oxidative versus glycolytic fibers. Some fibers prefer the oxidative approach, prefers the aerobic cellular respiration. These are going to have lots of blood capillaries nearby. They're going to have lots of mitochondria in the cells and lots of myoglobin in the cells. So that will make them redder in color. So they can allow for much longer periods of contracting because they have more energy being generated by cellular aerobic respiration. The glycolytic fibers prefer anaerobic cellular respiration. They have a larger glycogen reserve that breaks down to release glucose for anaerobic cellular respiration. They don't have as many mitochondria. They don't have as much myoglobin because they don't engage in that much aerobic cellular respiration. So they allow for contractions that are short in term. They can only last for a little while, and then they run out of ATP. So these two categories gives us our three types of skeletal muscle fibers. It's a mix of fast slow twitch and oxidative and glycolytic. So here you can see this lovely image of a skeletal muscle that's been transversely cut. You can see the lighter color, the slightly darker color, and the very dark color. So we have the slow oxidative fibers. Slow oxidative fibers are very red because they are oxidative. They are slow twitch, so their contractions are slower and less powerful, but they can last for a long time from all the energy generated by aerobic cellular respiration. So you can have long contractions without fatigue, but not as powerful. So these might be the fibers most important for, say, running a marathon or keeping muscles for a posture constantly contracting. We have fast oxidative fibers, fast twitch, so they produce a faster, more powerful contraction that doesn't last as long. Oxidative because they do have myoglobin and mitochondria, but not as much as the slow oxidative fibers. So they are a bit lighter in color. Some might even call them pinkish in color. These would be good for, say, long walks on the beach, exercises that need to go on for a little longer, but don't need to be super, super powerful. And then finally, we have the fast glycolytic fibers, fast twitch, so very powerful, fast contractions. Glycolytic, they're using anaerobic respiration, so they're whiter in color, and they can only work for short bursts. So very strong, but doesn't last very long. So for instance, if you're sprinting to catch the bus that you're fixing to miss, you might be using a lot of fast glycolytic fibers. Powerful run, but only for a little while, then you get tired. Red, pink, white, slow oxidative fibers, fast oxidative fibers, fast glycolytic fibers. And in our skeletal muscles, we have a mix of all three, but the ratio between them can vary from muscle to muscle, can vary from person to person based on genetics. So some muscles that need to act quickly, say if the iris of the eye would have lots of fast glycolytic fibers. Our posture muscles that need to contract for long periods of time would have lots of slow oxidative fibers. That is where we will stop with this part of the lecture.